All right, everyone. Good day, everyone, and you're welcome to the third webinar series on the HackBio to the World series. I'm Fatima Atoyin, and I'll be your host for today. So, HackBio to the World series came about because we understand the problems and the difficulties that people have in showcasing their work or in talking about their work or applying for the next things to do in life so they can achieve their goals. And we decide to come up with this series where we'll invite past interns from HackBio to talk about their process, to talk about how they've been able to scale up, to give us the good and the bad and the ugly side of the application process, because it's always good to learn from another person's mistake. And this is the third series. The first series we had Miss Titilola, who talked about how she was able to scout for the application, and she made us understand that it is not the big schools that you can always look out for. We have some other schools that you can apply to and they would give you better offer or edges. And then last month we had Michael. Michael talks about how you can apply your statement of purpose, how you can answer your interview questions, and how you can talk about your skill when you're asked to talk about the skill you have in bioinformatics. Now, today we have someone else who is going to teach us how you can work, how you can start your projects from the beginning to the end. Because in the survey that we sent out, a lot of people said they do not have they have little or no what's it called, knowledge on bioinformatics. And I think this is actually a good one, because if you're on this call now, you will learn the stages, how you can start and how you can go about it. Now, we also have people saying that they don't know how to showcase their work. They don't know what to show. They don't have a portfolio. When by the time we're done with this score, you would be able to understand how you can showcase your work, how you can add your projects to your portfolio, and how you can talk about it. And that is why we have this guest speaker with us. But before we move to the guest speaker, I would like us to know what HackBio is. What's the idea behind HackBio? Why is HackBio HackBio? And to take that, I would like to invite Richard to give us a little bit about what HackBio is. Richard, are you there, please? Richard, can you hear me? Oh, okay. All right. You can unmute yourself. Yes. Please, can we give virtual class to Richard as takes us on a little bit on the history of what HackBio is all about? Over to you, Richard. Thank you. Richard, you can start talking.
Hello.
Yes, yes. Hello, we can hear you.
All right. I would like to open my camera and see. All right. So it's a nice opportunity to be here with you guys, and I'm going to talk a little bit about bio. Yeah. So for those of us who don't really know who we are, to get a little glimpse of it. So HackBio is an organization founded by Adewale Ogunleye. Sorry if I don't mention the names right. Yeah, so with a motive um, we do so by providing hands-on projects and mentorship to individuals who are interested. So it cuts across. So it's for life scientists and computational or computer scientists. So we provide um, courses and on demand. Like we have courses on our website where you can enroll, and also we have mentors available during specific time, during the time of internship. So you can also enroll and you get mentorship, guidance, and all that. So as we speak now, we are currently on the AI in genomics internship, which started off 18th of this month, and we are helping interns or people who are interested to get the knowledge and move forward. Um, Richard Ajikun, as you can hear me, I'm also um, a past mentee or an intern from HackBio. And a lot of us are out there, and today, as Fatima said, we'll be privileged to listen to one of them. Yeah, so with HackBio, we have our website where you can enroll in the courses. We have the pathway course where you can, the videos will be there, or and then there on demand where you can follow through structured courses where you end up building your skills in bioinformatics. So all you need to do is to sign up, and also you can request for one-on-one mentoring and everything. So without much ado, I'll leave the floor back to the host, Fatima, to continue with the work, and as we listen to our brother Samuel. Yeah,
Thank
thank you.
you. Thank you so much. So thank you so much, Richard, for that. And like you said, with HackBio, you get to have lots of skills because as we know, bioinformatics is like a relatively new field and it's rapidly evolving. And because of the fact that it's constantly changing in HackBio, we have this thing that's every internship, we tend to switch the theme of the internship to meet with the current demand of the society. And like you said, we started the new internship on the 18th of this month, which is AI in genomics. And that will run for roughly 8 weeks. And moving on, I would like to ask the guest speaker to get ready so he can teach us, because that's what it's practically doing today. It's teaching us how we can start our workflow from start to the end, how we can use it, how we can add it to our portfolio, and how we can showcase it. So Samuel Anifowose is currently a PhD student, and I will not be saying much about him. I'll give him the floor to talk about himself. I want him to brag about himself. So Samuel, are you ready?
Yeah, I think so. Thank you.
You're welcome. All right. So please let's
Um,
welcome Samuel. Let's welcome Samuel so he
Thank you.
can. You're
All right.
welcome. You're welcome.
Yeah. So I believe you can all hear me, right?
Yes, yes.
Yeah, that's cool. So it's nice to be here. I have few of my mentors here actually. So I'll be careful with the way I put things here. So first of all, I sincerely want to thank everyone, Mr. Wale, Mr. Richard, and Miss Fatima for having me to talk to the community. It's a great privilege, and I do not take it for granted.
You're
So
welcome.
let's dive straight into what we have for today. Just let me know if you can see my screen and continue from there. Yeah.
It's still loading.
Oh, okay.
Yes. Should be up in a bit.
Makes sense.
Please, if you can see someone's screen right there, you can just send
Just
a
let me
message.
know if you can see my screen. And
I think it's still loading. Yeah, it's still loading. All right. It's up now. Someone can hear me?
yes, I can hear you.
Yeah, the slide is up now. You can start.
Okay, that makes sense. Okay. So just for emphasis sake, because of the time, so I might not actually be diving too much, like I might not be diving so much into the technical aspect of this work, and it will be a good one because of the time, so I can actually manage the time. So first of all, I will start with this. Um, when you are setting out to understand what you really want to do, and you you need to put together your skills, your knowledge, and all of that. So this is where I would say that you should start from. You already know your area of expertise, or where you intend to, walk into, like where you intend to research, where you intend to be in the nearest future. So what I would say that you should start from is start laying your hands on some papers in your field. What do I mean? There are a lot of people that are doing a lot of stuff in your field, in your area. So start reading papers about them. If you're considering traveling to the UK, to the US, or to any part of the Europe, try looking for a specific paper from the lab you intend to go. I don't know if that makes sense. Like the institution you intend to go, read papers on them, check the gap. You already have the bioinformatics skills. You have the necessary computational skills that you want to put into place. So start reading up the papers. A lot of them are generating absolutely so much data in their labs, and that's a fact. A lot of them are generating a huge number of data from their lab, and they actually do not know how to bring in bioinformatics. Let me put it that way. Don't let me be don't let me be too complex about that. Let me just put it that way. So they need more hands in the place of bioinformatics, in the place of analyzing their functional genomics data, their omics data, and the likes. So I would say that is where you need to start from. So once you're able to understand the gap, you're able to understand where you can come in as a bioinformatician, then you can be able to send them a mail. This is what I've been able to notice and all of that, and that seals it. And hopefully you should be able to get it. So for the purpose of this webinar, I would not be diving too much into the technical details, as I've said. I will just mention some of the tools involved, and I will also focus on five major areas that will bring about the story behind the projects that I'm doing and how I got into it, actually. Um, and then uh, the five major points that I'll be mentioning today, after I share my story briefly, is the biological question we are trying to identify in the lab where I'm working, the way we are generating the data, the workflow architecture, the output we have presently and the broader impacts that I personally am putting into place for the lab to be able to use and for the wider global scientific community. So these 5 points actually captured my the workflow itself, and it captures the work itself. So let's go in. Yeah. So how many of us know this guy? I don't know if you all know him, but this is William Edwards Deming. He's an American statistician, industrial engineer to be precise. And one of his famous ideas was simple but powerful, and that was in 1989 when he came up with the Deming management method. And he was able to say something which I was able to quote, and this word has actually stayed with me the very first time I saw it. He said, "In God we trust. All others must bring their data." And that's the essence of where we are today now. And in addition to that, I also read something about Drake, and he said sometimes it is the journey that teaches you a lot about your destination. That is absolutely fine. The more you keep going, the more you understand uh, where you are going. The more you explore, the more you get to know what you need to add to yourself, what you need to put together in order for you to achieve and to get to your destination. Right. So, quickly to my journey, I began from zoology, actually from Obafemi Awolowo University, and initially I was interested, just like every other young um teenager going into the university. I wanted medicine. I wanted to be in human health and all of that. But I got into zoology, and eventually I started with vertebrate biology and then proceeded to my MSc, where I did molecular parasitology at Federal University of Abeokuta, Nigeria. And we took a module on neglected tropical diseases where we extensively looked into different parts of neglected tropical diseases. I'll be saying a brief introduction about that aspect later. And towards the end of my MSc, uh, I was able to get a grant to do a particular work. My MSc project was on lymphatic filariasis. And then towards the end of that MSc, I was able to get a grant to work on soil transmitted helminths in the whole of Oyo State rural environment at the time, because they are actually endemic groups at the time. And after the time when I completed the grant as the principal investigator, I I then knew that during that project, actually, I knew that there was more for me to explore. There was more for me to know beyond molecular identification, beyond molecular characterization, and all of that. So I I started venturing into knowing what it takes for me, and eventually I came into the knowledge of bioinformatics. And when I knew that there was something called bioinformatics, I finished up the principal, I mean the project, and I submitted my technical report, and I started a lot of training in computational biology, in bioinformatics, and I started from paying for some trainings at the School of Biological Sciences at the University of Cambridge in London. And I started with Python and R. And after that time, I came in contact with HackBio. At the time, I've never started doing anything with them. I was just seeing them, all around, and I was taking my time because I did not just want to follow what of the trainings also on Coursera, and I was able to sit down and understand the trajectory of how my skill set should go. And then I started with the HackBio with the internship on data science for life sciences using R. And that was how I started with HackBio in 2023. Yeah, 2023. And from there, I've been able to build my bioinformatics skills through independent training, and of course, just like I've said, from HackBio. And presently, I'm in Newcastle University in the UK, studying biomedical informatics, and I'm developing different kinds of expertise in precision parasitology for African populations. And that's a brief idea of what I'm into. And I'm presently into computational biology with focus on neglected tropical diseases. Right. So, what are neglected tropical diseases? This is actually a diverse group of conditions that affect people in tropical areas. Many of us are familiar with them. They are not only restricted or limited in Africa, they are across the globe, including the Americans, the Asia and all of that. And some of the examples, some people usually feel that NTD is just about maybe a particular pathogen in a particular um, specific for a particular group. But let me say that NTD cuts across all the pathogen groups. We have them as the parasitic entities, we have them as bacterial entities, we have them as viral entities, we have them as fungal entities, and even some non-infectious diseases such as the snakebite envenoming, which is actually a potential life-threatening disease, and it is caused by toxins. And this is actually injected during the bite of a venomous snake. And in fact, as I'm talking to you, there are a lot of grants concerning this particular aspect because it's leading to having over 150K deaths annually. And it is mostly concentrated in the rural tropical regions. So apparently, there are some data facts about neglected tropical diseases, and globally, over 1 billion people are affected by neglected tropical diseases, with 1.5 billion requiring interventions annually. And in Africa, 40% of the global NTD burden is found in Africa, actually, and it's making it the most affected region. And in Nigeria, of course, this carries one of the highest burdens in Africa, with millions at risk of diseases such as schistosomiasis, onco, STH, soil transmitted helminths. And the reason why we are having an increasing risk is because of the recent stop in the ODA, the Official Development Assistance from the US. So there is a increasing level of infection, of risk infection across the globe right now. And the majority of these neglected tropical diseases do not have an effective drug that is being used to, take care of this infection. So there is need for different drugs to be developed, different biomarkers for specific therapeutics to be developed across the population. And this is why there's need for precision medicine, targeted drugs for a targeted population, right? So majorly, my own work is because in the lab where I'm working, we make use of cell lines, and we literally do not use the human clinical samples. But because of the fact that I am concentrating more on African population, so we are looking around getting samples from Nigeria and a couple of other African countries where we'll be able to ascertain the solutions and the results that we are seeing. And this is what led into me looking into neglected tropical diseases with my skill of bioinformatics and computational biology. So um, at the moment, they've generated a lot of data, which it's actually very important in the lab. And they generated some of this data through some of the skills, through some of the technologies that are actually advanced in the place of research at the moment, which is IEEE technologies, and part of which we have the single cell transcriptomics, the bulk, the spatial transcriptomics. In fact, as I'm speaking now, we are conducting spatial transcriptomics library preparation in our lab, and that makes it more interesting. Yeah, let me put it that way. And these are some of the things that we do in the lab. We do proteomics, genomics, chromatin accessibility, and single cell RNA sequencing, transcriptomics. I mean, all the transcriptomics itself. So we are particularly involved with kinetoplasts, which are the trypanosoma and the leishmania. Now, let me say this before I go on. I was able to get in contact with the head of the department. She's a doctor, and I was able to get an access to her through a professor that I met on LinkedIn, if that makes sense. So, for people that do not, or you have not actually come to the place of optimizing your LinkedIn account, I mean, making yourself visible and all of that, and not even just visibility, making the right networking. Yeah, let me put it that way. Networking via LinkedIn. It is very, very important. I got the access to this lab through a professor that I met on LinkedIn. And the professor is not in Nigeria. You understand? So I just sent the message to him. I spoke to him about what I intend to do, my vision, what I've done, because at the time I co-authored a paper on a grant as well. And the grant that I also won, that I wrote the technical report, I also sent it across to him so that they will be able to see what I've done so far. And eventually, it linked me up with the head of the lab where I am at the moment. So that's what we are doing. And we are working with the kinetoplast, the trypanosoma, and the leishmania at the moment. And these are parasites that are responsible for severe disease. They are under the protozoan neglected tropical diseases. And one of the things about kinetoplasts is that they have a very intriguing cycles. I use the word cycles because they have life cycle and they have cell cycles. But presently, because of my mechanistic ideology about cell-to-cell interaction, I wanted to study their cell cycle, and this is why I was asked to come into this area with my bioinformatics analysis. And presently, I am focusing on my ability to understand the parasite cell cycle. And why am I doing this? It's because we are trying to understand, or let me say, we are trying to identify the proteins that influence the cell cycle of this parasite. If we're able to identify the proteins that influence the cell cycle, then it will give us or let me say, it will contribute largely to improve the drug development therapeutics. I don't know if that makes sense. So if we are able to understand the proteins that guides the cell cycle, I don't want to go through the life cycle because it will just take so much of our time. And that's why I'm just sticking to the cell cycle. And I want to be very fast considering the time that I have because of what I have done so far. So understanding the proteins, as I've said, understand the proteins that influence each stages of this cell cycle will make us to be able to contribute to improving the drugs development, the biomarkers that are necessary, because this kinetoplast, as a matter of fact, does not have any effective drug and is one of the leading neurological complications. And at the same time, it causes organ damage, it causes disability, and in so many cases, it leads to death. And there is a very limited treatment option for the kinetoplasts. They are a family and it's very difficult. So, and for us to be able to understand that aspect, we needed to start something. And that was when I came into the lab. So right now, this is a workflow that has produced a promising result in the lab. As a matter of fact, I developed this workflow after I think HackBio did the last internship that was done before this present one that is ongoing is single cell internship. And I took my time to understand that. I was part of the internship anyways. I took my time to understand that because I knew that that is what I'll be doing. Also, I also looked up to some resources online for me to understand that. And eventually I came up with a workflow that befit our uh, work in the lab at the moment. Please, I want to be sure if you are following. I need thumbs up. Just give me a thumbs up if you're following wherever. Just give me a thumbs. Fine, awesome. That makes sense. So this is a workflow that I actually developed in the lab, and this workflow has produced promising results. In fact, as of now, we now have people from other labs that are interested in this workflow and they started making use of it, especially those people that are interested in other pathogens related to neglected tropical diseases. Let me put it that way. So, to make this analysis reproducible and reusable, I mean, across scientific communities, I didn't just want it to, since it has started producing results for us, I didn't want it to be limited in a way that we are the only ones that are making use of it. So I thought of translating the workflow into a kind of modular pipeline, and the best pipeline to use or the best approach or method to use is through Nextflow. Then this is where I'm going to come in. If you know you really want to increase your ability of bioinformatics skills into more of advanced computational biology, there is no how you must not. In fact, let me put it this way: you should learn how to reproduce your workflows. And let me share one of the beautiful reasons. The reason is that once you are able to produce it, and define it in a reproducible way, you have given a lot of scientific community in your field, in your area, to be able to make use of your work, of what you have done. So it gives you more feasibility, it gives you more authenticity in your field, in your area, and it also gives you more recognition in your field. I don't know if that makes sense. So there is no how you won't do without Nextflow, NickMake, but for me, I enjoyed making use of Nextflow. So I was able to build my analysis and translated it directly into using Nextflow, following 6 major steps, which I'm going to highlight now. Yeah. So I splitted the analysis into modules. For emphasis sake, when I was building the workflow, I could not use all the data that have been generated. And let me start with saying this. All the data that were generated were from single cell data sets. After they've done the wet lab, they've done the single cell library preparation and all of that, then we brought in, I was not there. So they brought in the each of the cells. Now let me say that each of the cells were tagged with a particular protein tagging fluorescence, which they called amnion green. So after tagging each of these cells, don't forget I said they are single cells from the cell lines that were processed. So after tagging these cells with these proteins, with this fluorescence, the amnion green, then this amnion green would help us to identify them through bioimaging, would help us to identify each of the cells, and at the same time would help us to identify, because they have been tagged, so it will help us to identify the proteins that are available in each of the cells. I don't know if that makes sense. So if we are able to identify that a cell is having a high intensity of a protein, it means that that cell is being driven by a particular protein. Now, for clarification, the cell cycle has 3 stages. We have the early stage, the middle stage, and the late stage. In trypanosoma, in kinetoplast, so we usually refer to the early stage as the 1N, 1K. 1N means that it has 1 nucleus and it has 1 kinetoplast. That's the early stage. Then it moves to the middle stage. In the middle stage, this is where the kinetoplast, which was 1K, it was just 1, gets divided into 2. So in the middle stage, we now have 1 nucleus, 2 kinetoplasts. Then from the middle stage, it moves to the late stage of the cycle, where we now have a duplication of the nucleus. So instead of us having 1 nucleus, we now have 2 nucleus and 2 kinetoplasts. So the stages now moves from 1N, 1K, which is the early stage, to 1N, 2K, which is the middle stage, and then to 2N, 2K, which is the late stage of the parasite. Now, we are now trying to understand the proteins that express each of these cycles, of these stages. So in order for us to now understand that, we know that the proteins are available, but we want to see where their intensity lies. For example, in the early stage, do we have the proteins are intensified in the whole cells, or in the nucleus, or in the cytoplasm? So if we are able to understand the dynamics of this, it's just like saying you want to understand the gene expression across a particular disease state, infection state, whatsoever. So we are doing the same thing for protein now, to understand the dynamics of the proteins across each of these transition in the cycle. So after doing all of that, I'm just trying to be very fast because of the time. So after doing all of that, I was able to use this workflow for that process. And then I connected to the next flow. And in the next flow, this is what I'm about to talk about. So I started the analysis using R. I wrote all my codes and all of that in R. And for me to now bring that analysis, the pipeline, into Nextflow, I needed to divide them into modules. So this is the first thing that I did. I split the analysis, the scripts. The analysis into modules. So for example, I started with pre-processing, loading your packages and all of those things. And then you go on with different kinds of stuff. So you do your quality control, you do your filtering, you do you continue with your PCA, you continue with your UMAP, you do your trajectory analysis and all of that. So I had to split this analysis into modules, because so that Nextflow will be able to understand. In Nextflow, you have to have your process and your workflow, and your work itself, so that Nextflow will be able to run that analysis accordingly and sequentially. Right. So after splitting the analysis into modules, I define the processes in Nextflow. So the processes are, for example, the pre-processing aspect of the analysis in my R. I took it out, created a new script for it in R, make it one of my processes. The aspect for I'm going to go into that a bit. The aspect for the PCA, the UMAP, I brought that into another script, add it into my Nextflow. The aspect for Slingshot, these are normal single cell algorithm, step-by-step pipelines. So the trajectory, I also did the same thing and all of that. So I connected all of them together in the main.nf. NF is Nextflow. So in my Nextflow, all of those things I use VS Code, Visual Studio Code. And this made the work so seamless for me, right? I don't have to go into R and leave R and come back into command line, so I can easily have my work done in Visual Studio Code. So that was where I was able to do all of this thing. So I connect my processes in main.nf. I manage my inputs and outputs. I validated my reproducible results. Now, don't forget, I have already done this analysis with a subset of the data in my R. So I already have the plots. I have my PCA plot. I have I'm going to show you some of the plots that I've been able to generate. So I already have those plots. My pipeline was working so well. So all I needed to do was, once I brought all of these things into Nextflow, into Visual Studio Code, I was able to validate the results compared to what I had from my R Studio. I don't know if that makes sense. So that makes me believe that what I've done with my Nextflow in the Visual Studio Code was exactly the same, and it yielded exactly the same result that I got with my subset of data. And lastly, which I have not done, which I'm hoping to do, and this is one of the reasons why I participated in the last hackathon that was done by Nextflow in the month of March last month. I'm preparing to submit the Nextflow workflow, the pipeline now, to NF Core, the Nextflow core itself. And the reason why I'm doing this is because it will serve a major breakthrough, one, for African population. So anyone interested in this kind of work can take this workflow. You know, if you like, you might just add one or two things to it, depending on your preferences. You can take it, put in your data, run the analysis, and you will get the exact same thing that I got from my work, right? So yeah, this is what I did. These are the modules that I built in the Nextflow. The preprocess script. I have the preprocess script. I have the PCA and UMAP script, the slingshot script, and I have the plot result script. So all together, I bring them together and put them in the main.nf. Because of time, I would have loved to, show us all of that. But if you want to know more about this, you can always reach out to Mr. Wale HackBio. They will always reach out to me and we will discuss, or you have a work around something like this, we can discuss further on this. And then I was able to create the Nextflow process, as I've said, and we're able to plot the results. And from there, I was able to manage the inputs and outputs. I also process each of them to configure the receive input files to generate the output, and lastly, to pass output to the next process. And the reason why I did this aspect is because it will ensure that the workflow remain reproducible and traceable. Any work, any analysis pipeline that you built into Nextflow and no any other researcher could make use of it, then it doesn't have any essence, right? So it is important that the input and the output in the Nextflow block codes that you already have are reproducible and can be traceable. Someone might want to implement what you have built, and that's the beauty of this workflow and getting involved in hackathons. So yeah, let me put it that way, because that was one of the first things that I learned from the hackathon itself. Then yeah, that's all it. Yeah, so because the work is still ongoing, I just picked some of the plots that we already have, and the first one is our PCA. So this is the first plot that we got. This is the PCA that I got, and this PCA just talked about all the cells that were eventually generated were over 500,000 cells with about 60 features of the cells. And this PCA just compressed them put them together in clusters that we now have just so if you can look at it, we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So we now have all of them in 10 futures. So this makes us to develop our correlation map. And this correlation map, I'm sorry, it might be very small, is because I dedicated this slide alone for the plots because of the time. So this correlation map actually showed that we have the strongest part. If you can see the colors, we could have some in dark colors, darker than other ones. So it showed a very strong variation across each of the features. On this side are the cell features, right? And on this side, these dimensions, they are also the PCA, right? And after getting all of this, and we're able to understand the PCA itself and the variation across the cells, we're able to use the PCA generated to develop our 2D UMAP. And as you can see, just like what I said, this is the cell cycle of the trypanosoma parasite. So it starts from here. All of it, I've labeled it as 1N1K, 1N2K, 2N2K. So the 1N1K, which is the early part of the cycle, they are the ones in yellow. So you can see that the cell starts right from here and keeps continuing the trajectory until it gets to the middle, which is 1N2K. So the 1N2K now, that's the middle stage of the cycle. And then lastly, it transitions into the 2N2K, which is the last stage of the cycle. These were generated because the data has been filtered. I filtered the data, the quality control was done, and we're able to see the trajectory of the cell cycle, and which actually look very nice and beautiful. And for F, okay, let me add this. Since we already have the 2D version of the the UMAP of the cell cycle, I now created the 2D of the UMAP too, but this time around as clusters. And the reason why I created this as a cluster is because we needed to know which of those cells out of the subset that I've used before earlier to build this pipeline. I use over 20,000 cells to build the workflow. So for me to understand the way the trajectory is going, I need to understand where the starts cluster, the starting cell starts from, and where the late stage ends. So for me to do that, because if I'm drawing this slingshot trajectory at the end of the day, in the slingshot function, there's a place where you need to impute your starts and your end cells. And by the time you do that, you'll be able to add up to your stretch and to your shrink. And eventually, if you look at these clusters, these are the ones starting the cells, this site, and this is where the cells are ending. So this is the start point of the cell cycle, that is the early stage, and it proceeds. These are all the cells. If you look at this to the showing the UMAP, you would see the yellow starting off on this side to this side. So this is showing that all the cells available here, they're the ones responsible for the early stage. And why you could see the green ones here, you will see that these are the cells that are responsible in the middle stage, and lastly, the ones responsible for the late stage. So these last cells and these early cells were the ones used that were able to draw the slingshot trajectory. And you could see how smooth the trajectory went. Yeah, so yeah, and for this, I also have, this is showing the intensity of the proteins in the cells. So this plot is just like the total events going on in the cell. Yeah, let me put it that way. So the total events, and when I say event, I meant to say that the dynamics of the proteins in the cell. Yeah, the dynamics of the proteins in the cell. So, as I've said earlier, the yellow ones represents the early stage, the green ones represents the middle stage, and the blue ones represent the late stage. So you could see that in the cell, the protein intensity increases over the pseudo time. The pseudo time is more of these are the dimensions of the stages of the cell cycle, right? Yeah. So uh, we now have a knowledge of the proteins moving across and transitioning from one cycle, from one stage to the other and to the last stage, right? And this is all for the ones happening in the cell. Currently, this is the ones in the nucleus. As far as this work is concerned, you still also have to be sure that what you are having is in line with what literature has said about some of these proteins. There are some proteins that have been identified from literatures that they govern or they influence the early stage. Some of them include the CDC45, which are some of the ones that we also got. CDC45 is responsible in the early stage because they influence, or let me say they initiate DNA regulation replication. And of course, when you look at the early stage, DNA replication is important for the division of the nucleus in the cell, right? So we also have the cyclins and the cyclin related kinases. And these are all the proteins that we identified eventually. And with the way things are going, we're able to also. I'm not really deep into proteomics as that, but with what is going on now, I think I might be having a good interest in proteomics now because at the end of the whole thing, we were able to identify that there are actually some beautiful proteins that were identified, and it makes a lot of sense. And they could actually be a novel proteins that regulate some of the cycle. So let me keep that one for now. And these ones happens in the cytoplasm. So you could see that some of them don't actually have a stable intensity across the pseudo time, across the cycle. And what that tells us is that majority of the proteins based on their localization, either in the nucleus or the cytoplasm, happens in the cell. So majority of the processes, DNA replications, the dynamics of the proteins do not actually occur majorly in the nucleus and in the cytoplasm. They actually occur entirely across the cell. Across the cell. And that's why we have this transitioning in the cell. And lastly, we're able to have a heat map of what is going on just for us to see the trajectory. I'll be changing this color palette anyways, because if you are looking at it now, you might be thinking that yes, we have a light blue, we have white, and we have deeper blue at this late stage. So it was supposed to be more of two-dimensional color palette. That's what I'm thinking, so that it would give more meaningful ideology to anybody just seeing it, even without interpreting. But let me just interpret this. So this light blue is showing the intensity of the proteins. These are all the proteins that were identified. Excuse me. So this light blue is showing some of the proteins. It's showing some of the proteins and the intensity of the proteins that happen at the early stage. The middle, you can see the white ones. The white ones is showing how they keep increasing over time. And the dark ones, that's this late stage. So at the top here, you see the pseudo time, the color changing over time, and that's for you to understand that the proteins are regulating from early to mid and to the late, and that's what we have at the pseudo time up there. And this was actually done with me using set score for me to have a very unique dynamics of the proteins, because if I should use the absolute data itself to run this heat map, it will not make a lot of sense. Fine, the heat map will be there, it will generate a very nice heat map, but then there will be more intensity of a particular protein compared to the other one. So the intensity will not be equal to each other. So that is the reason why I had to use Z-score, because Z-score will give us a perfect dynamics of each of them in the beans. So these are the pseudo time beans across, and I have the early, the middle, the mid stage, and the late stage. So we can see that majority of the proteins are actually they influence the cycle at the late stage, the transition. Some of them peaked very late, while some peaks very early, and once they peak early, some drop because of that, and then we have a larger number of them increasing over time across this cell cycle process. Now to my take-home message. The same William said something. He said, without data, you are just another person with an opinion. And that's absolutely incredible. So without data, you might have different kinds of ideas and all of that, but data will make us see, just like what I said when I started, that the grant that I won was more for me to understand the molecular dynamics, molecular identification and all of that. But now, because of that, I knew there was more. And the more that I knew there was about the data, and that we could generate a lot of biological insight from data, not just in every other human disease, but also in neglected tropical diseases. And that is where I'm focusing on. And one, my journey has shown me that computational biology can transform how we understand neglected tropical diseases. That's my area of specialization. Two, through this work that I'm presently doing, it's not just only about analyzing protein dynamics, but also building tools, workflows, and also help uncover biological meaningful targets. Quicker, in quotes, uh, we are now coming to the place of generating data. We are coming to the place of making use of data to understand a lot of things. And this is the reason for precision medicine. We cannot depend on the data that have been generated from the overseas, from the Western countries and all of that for us. And this is the more reason why many of us up there listening to me, you need to venture into your area of specialization. Look at the gaps there. Look at what you can feel. Sorry. Look at what you can feel and look at how you can bring them into place. Then lastly, my long-term goal is to apply these approaches toward precision pathogen biology, developing data-driven solutions that are relevant to African populations where these diseases remain endemic. And that's the absolute goal of my own personal mission. So it's my own deal to make sure that African population can get their own targeted drugs for their targeted populations, and eventually we'll be able to have design drugs for ourselves, and that will make a lot of sense. So lastly, I'm building computational tools to identify all of this in infectious diseases, and that is what I'm doing. And I hope that will make a lot of sense for everyone out there. Thank you so much for listening to me. It's nice to see you all, and it's nice to share my research. Thank you.
Oh my God, that's that is amazing. Can we just appreciate Samuel for this wonderful, wonderful, wonderful presentation? This is such a nice one, Samuel. In fact, if
Mm.
not for the time, I was so locked in. It was my alarm that went off to actually remind me that, oh, it's almost an hour to go. Like we have spent an hour here. If not, we'll probably spend the rest of the day here because that was such an amazing lecture. Thank you
Right.
so much, Samuel. And
Yeah.
if you all agree with me, I think we should have another session with Samuel because this is like, this is so nice. And this was really, really um, thoughtful. It's an eye-opening session. Thank you so much, Samuel, for this
No worries.
presentation. And
Yeah.
I'm still saying it. We'll still have to call you back because we have lots of questions for you. We have lots of
Right,
questions for you.
right, right,
Yeah,
right.
thank you so much,
Yeah.
Samuel. We
Yeah.
appreciate
Thank
you.
you.
Thank you.
Yeah.
All right, everyone. Thank you so much for joining in. And I know we have a lot of things we want to ask Samuel. Unfortunately, we need to end this call because I think we have another session with someone else. Now, if you have any question, you can always send it to the contact@hackbio.com so that we can forward it to Samuel, and whatever he gives us as his reply, we'll forward it back to you if that is fine by us. I'm really grateful that you guys joined in. This is an amazing turnout. I'm glad that you joined in, and I hope that you've taken one or two things from Samuel's story. And lastly, I would like to say that you should be you, follow your dreams. If you don't actually get what you want, you can always build on what you have. Like Samuel said, we all had hopes of becoming medical doctors, but at the end of the day, life threw some things at us and we were unable to go into that line directly. And look at Samuel, he's thriving where he is. And on that note, I would like to say a big thank you to Samuel, a big thank you to all of you for joining in. And thank you so much, Richard, for giving us that talk on HackBio. Thank you, everyone, once again for joining. I am Fatima Atoyin, and I hope to see you in our next webinar. Thank you. Bye, everyone.